The DNA is transcribing to RNA. The RNA is translating to protein, amino acids and protein. Guys, at first we will have an overview. We will speak about amino acid pool and amino acid turnover. Again, we will speak on digestion of dietary proteins and about the amino acid catabolism. Just I'm gonna tell you a little about each of them. A little, not so much. And about the classification of amino acids, and finally about some metabolic abnormalities I'm gonna tell you. Because at first, we've studied that glucose is stored in our liver and muscle in the form of glycogen. The lipids is stored in our adipocytes as TAG. But we don't have any storage of protein in our body. We don't have storage of protein and amino acids in our body. No. We should obtain amino acids from diet, from de novo synthesis, or degradation of body proteins. Do you know about de novo synthesis? No. Imagine the protein is it an amino acid structure or not?
Yes.
It is. Guys, if all of these components gather together from the first, it is de novo synthesis. De novo synthesis means synthesis from the origin. Our body produces it. Okay? This is de novo synthesis. So, our body maybe should obtain amino acids from food, from de novo synthesis, or degradation and breaking down of body protein. Guys, any amino acid that be more than our body needs, it will rapidly degrade and rapidly breaking down. Okay?
If we suppose that an imaginary situation, our body stores a protein, what is the side effect?
Because of ammonia, the amino acids have a large amount of ammonia. Okay? If we had the situation that we can't store that, obviously we had a mechanism for that. Otherwise, because of the ammonia that we can see in amino acid structure, I told you that we have this ammonia is toxic. If it becomes free in the form of ammonia, it is toxic, especially for brain and every other part. And our body should actually use lots of energy for excreting that. That's why we don't have maybe the storage of that. Guys, it is an overview that I won't explain all of that by detail. You can see that amino acid had, again, that these are in lots of situations and generally was carbon skeleton. Am I right?
Yes.
Now, when the amino acid breaking down, we have ammonia and and we have carbon skeleton. Okay? The ammonia should excreted from our body as urea, or maybe convert to other compounds.
Okay.
And urea will go out from our body through urine, exactly. Through the urine. But the carbon skeleton have a complicated pathway. I won't concentrate on that at all. Okay? But if you study and have questions, you can ask me. And guys, I share the slides for Zainab and Ali Amr. And please ask them to share with you, okay? Okay. The fourth slide set. There is four slides. Guys, this was related to overview of metabolism of proteins and amino acids. Now, we have a fluid. We have amino acid all in our body. In cells, in blood circulation, in extracellular fluids, in every part of our body. And we call that amino acid pool. Amino acid pool. In our body, because of the nitrogen that there is in amino acid, we need a balance. We need a balance. The amino acid that we intake and the amino acid that we get out of from our body should be in a balance. Did you get the point?
Yeah, how that's possible? How this balance is happening?
I will tell you. This is so important. If our liver works properly and our metabolic pathway works properly, the high amount of nitrogen will excrete in our body in the form of urea. Okay? The balance occurs in this situation. And it is in a normal situation, but there are some situations that we call, it is nitrogen balance, okay? This is nitrogen balance, that you can see that excretion of nitrogen is equal to our consumption. Our body can excrete the input nitrogen in a proper way, okay? In the other situation, we have positive nitrogen balance. Where is it? Where the nitrogen consumption is higher than nitrogen excretion. This is a positive nitrogen balance. Where, for example, in pregnancy, a child that is growing up, it needs a high amount of nitrogen, and the consumption of that is higher than the excretion of that. On the other hand, we have some situation, for example, some trauma. Or maybe in some malnutrition. Do you know about malnutrition?
Yeah.
Like kwashiorkor, marasmus. Have you ever heard about that?
No, but the malnutrition is not good nutrition.
In African country, you can see it's not important to memorize the name of that. They cannot intake enough food, enough calorie, enough carbohydrate, and especially enough protein. This is a negative nitrogen balance. That nitrogen excretion is higher than nitrogen consumption. And they are not in a good situation. And about amino acid pool, guys, the amino acid pool is supplied by three ways. The first way is the degradation and breaking down of protein in our body. Okay? The next thing is receiving protein from the diet. And finally is the synthesis of that Thank God you know one thing.
Yeah, that Dr. Khalid explains.
Okay. We have the non-essential amino acids that help the production of other amino acids and essential amino acids. Okay? For example,
From sun.
for example, phenylalanine from tyrosine from phenylalanine, or cysteine from methionine. They are conditionally essential. Or maybe, for example, glycine help to producing other amino acids. And the amino acid pool can be depleted by synthesis of body proteins, consumption as precursor of some other nitrogen-containing molecules, or conversion of them to glucose, glycogen, fatty acid, and ketone bodies. Guys, this is clear. Am I right? In this situation, the amino acid is produced, the protein is produced, okay? The amino acid is produced, and in this situation, the amino acid is consumed. When the amino acid is consumed, when they consume for synthesis of protein, we don't have amino acid anymore because it comes to structure of proteins. Am I right?
Yes.
In the other way, maybe the amino acid convert to other nitrogen-containing molecules. Do you know other nitrogen-containing molecules? You know, I'm sure. Have you ever heard about nucleotide
Yes.
in DNA structure?
Yes.
They contain nitrogen. And that's why some amino acids, like for example aspartate or glutamine and glycine, are used for nucleotide production. Okay?
Yes.
Did you get the point?
Yes.
Then maybe some amino acid used for production of other nitrogen-containing molecules. And on the other hand, maybe amino acid convert to glucose, glycogen, fatty acid, and ketone bodies. I just will name that. I want to explain you about the mechanism of that. Guys, every protein have a half-life. Do you know about the half-life?
Yes.
What is that?
It has a cycle, produce in specific time, and after that the body discharge from it. That's what I understand.
The time that need to become a molecule in half amount of the origin. For example, here we have 4 grams, for example, albumin. Okay? After 2 hours, the half-life of albumin is, for example, 2 hours. After 2 hours, it would become 2 grams. 2 grams. After 2 hours, it will become 1 gram. It is half-life. Did you get the point about the half-life, about everything, about radioisotopes, about nuclear products like uranium and every other thing? We have a half-life.
Yes.
And in medicine, you have, for example, you go for scanning, and they produce and they inject some iodine. Actually,
For fluoroscopy or
for example, for having a clear picturing, for example, from a tumor,
yes.
they inject some radioisotope product, and they, for example, tell the patient in this time you shouldn't actually interface with pregnant women, with children, because of the half-life of those products. Did you get the point?
Yes.
Guys, did you get the point about
have half-life. It's mean after 1 hour or 2 hours will be synthesized into half weight or half. It will become to the half. We have these elements when we want to take X-ray or fluoroscopy or something like that. They inject some elements in our body, and they give us a period of time that we should not mix with pregnant women or children. And after this period, this element will synthesize in our body, and because of the half-life, it will
and excrete from our body. For example, they told us that I give you, for example, 2 grams iodine, and based off the half-life of that they know, they call me, for example, after 8 hours, it will be clear from our body. Okay? It is related to half-life. And if the dietary protein be our needs, they will convert to glycogen and tag and store in our body. Could you explain about half-life to your friend? And this is related to the synthesis and degradation of proteins. For example, we have an enzyme that do, for example, glycolysis. The half-life of that should be a limited time. They can't work completely and continuously. It should break down. It is related to half-life.
I should explain.
Is it possible? Thank you.
كل البروتينات في النظام احنا قلنا البروتين ما يتخزن بالجسم، لان البروتين اذا تخزن بالجسم راح تزيد نسبة الامونيا. هذا مرينا، فالبروتينات شلون جاي تشتغل بالجسم؟ تشتغل بنظام اسمه الحياة او هيلث لايف. زين هذا شنو هو النظام؟ تقول هذا النظام اشبه بانه تذكرون اخذنا بالاشعه انه بالاشعه يعطونا فد مواد او شو اسمه يسرق فد انجكت للمواد داخل جسمنا. هذه المواد راح تخلي هاي المنطقه اللي احنا نريد ناخذ لها اشعه برايت اكثر او بها مواد بها تكون ملونه اكثر. هذا الشيء زين هاي المواد احنا من اخذناها لازم الجسم يتخلص منها. شلون الجسم يتخلص منها؟ يعني هذا من ساعه ضربه مثال قالت 4 جرام ورا ساعتين تصير 2 جرام، انه الماده تتحلل داخل الجسم تنشطر الى 2 الى ان يتخلص منها الجسم بعد 8 ساعات. البروتينات هم شيء عندها فد وقت محدد الجسم يتخلص منها، فد مثلا 8 ساعات، كل ساعتين راح ينشطر. مثلا انت ايش قد اكلت بروتين؟ 2 جرام، هاي راح تتحلل لمده 8 ساعات، كل ساعتين راح تصير هكذا الى ان الجسم يسوي له اكسكريشن.
Did you get the point? Are you good?
I'm good, but a little bit tired.
You're sleeping?
No.
Guys, it was related to protein turnover that we need in our body. And this is the summary of all of the things that I've explained in the previous slide. Guys, this is a human just with stomach and intestine. A big intestine and no other muscle and other things. Guys, we want to have a journey with our food to our intestine, and we won't continue our journey till the end. It's not a good story, okay? Why? Because it will enter the stool, yeah? I'm telling we have a journey with our food to GI system, but I'm saying that we should end our journey in the middle part, not till the end. Okay, guys, at first we have proteins. We have proteins in our diet.
Okay.
Is it need to, what did I say?
I told them that please pay attention.
I told you about this. Could you tell me in English about the journey?
No, I
start from the stomach. From what? At first. In gastric secretion, we have acids and some enzymes. Which enzyme? Pepsin and HCl or hydrochloric acid. These components help to breaking down the proteins to some peptides. Guys, at first, do you study about proenzyme or zymogen? Please.
We understand what's coenzyme. It's the enzyme that need another thing to be active.
And do you know that in some situation the enzyme should be inactive, and in some situation it needs to activate? In GI system or in gastrointestinal tract, enzyme at first should be inactive, especially protease. Imagine the protease be active in every time. Is it good, for example, the enzyme be activated in our small intestine? After a while, it starts to actually degrade all of the protein in our intestinal wall. It should be inactivated and become active in some situation. I will explain you. Every time that I say I will explain you, I remember yesterday. She says just I will explain you, I will explain you. I cannot understand nothing. It is great. It was great. But I promise I will explain you. Guys, at first, the HCl and pepsin
Will digest the protein to peptide.
yeah, two peptides. After that, the pancreas again start to secrete what is it? Do you know what is that? Bicarbonate. Bicarbonate. It is basic, a powerful base. We have acid and base. The bicarbonate is a powerful base in our body. Okay, this is in pancreatic secretion. After that, we have some enzyme, trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase A and B. I will explain you. And after that, these enzymes become activated and breaking down the peptides to D and tripeptides and amino acids. Where we are in intestinal lumen. As Alice said, it is the empty area of our intestine. It is an empty tube. Okay? After that, what is this? Enterocyte, exactly. Now the amino acids and di and tripeptides enter the enterocyte, again degrade here by enzymes and convert to amino acids. Now amino acids go to blood circulation to our body. It is the whole story about the protein digestion. I think it is easier than others now.
Yes.
And about the enzyme that I promised you to explain. It is the protease.
This diagram for the enzyme that prompts us to explain.
Yes.
We are so friendly if you don't explain this. We don't want.
No worry about these structures. No worry about that. Just about this part. I will explain you just about this part. Okay? It's not important to memorize these. Even for Iranian students, I tell all of them it's not important for memorizing too. Each amino acid is affected by enzyme. I want to explain you. I told you that we have trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase, They come from pancreas secretion to the small intestine.
Okay.
From pancreas secretion. They are inactive.
Okay.
They release from pancreas to the small intestine and they are inactive. They should activate. How?
When the protein comes to the intestine.
Yeah, by an enzyme under the name entropeptidase.
What?
Entropeptidase.
Okay. The sentence, repeat the sentence.
It is in the small intestine. When the pancreas secretes the enzymes to the small intestine, from here, the pancreas secretes the
Just play on the screen.
what's happening here. Okay.
Click on screen.
I am rural in this situation, you know. In Iran, we don't have these smartphones. Guys, the pancreas secretion have these zymogen. Okay? These are inactive.
Okay.
They should become activate. How?
Why intro?
We have entropeptidase in the small intestine. At first, breaking down the trypsinogen to trypsin, and this trypsin starts to activate again trypsinogen, activate chymotrypsinogen, activate proelastase, activate carboxypeptidase A and B. How? By breaking down a small part of protein and convert that to an active enzyme. You can see maybe the pro or ogen. These are related to the zymogen proenzymes. Did you get the point? When you see pepsinogen, trypsinogen, chymotrypsinogen, they are zymogen, they are inactive. Or maybe you should hear proelastase, procarboxypeptidase, proinsulin, pro-PTH, about everything. They are inactive. The pro part should cut and convert to an active form. Did you get the point?
Yes.
This is related to the pancreatic enzyme. Guys, do you need your friends explain in Arabic or not?
It's easy.
It's easy. Okay, all of you. Okay. Thank you. This was related to cleavage of dietary protein. Now, guys, this was related to amino acid that enter to our blood circulation. They go to muscles, they go to, for example, in my eyes for producing some proteins, they come to my liver for producing some enzymes, then it comes to my, for example, my muscle hand for movement, for every part of our body, this protein is used. Now, it is used and now it is the time that amino acids should breaking down. For any reason, I don't need that anymore. What is happening? We have some common reaction that it is related to the removing of alpha amino group and have some specific reaction that it is related to catabolism of carboskeleton. I won't explain it to you. And other things, we have urea cycle, just I mentioned a little bit for your understanding and know about the mechanism of urea. Guys, this slide just tell you, it's not important, know about the mechanism. It is just telling you that the nitrogen is dissociated from amino acid by by these way, transamination or deamination. It's not important to memorize, just I wanna that in this situation, amino acid have carbon and ammonia. Am I right?
Yes, the ammonia go with urine.
Yeah. Now, at first, the ammonia should be removed by this process that it doesn't need you know about the details. And what is happening in the next? In the next, guys, the most important thing that you should know, the ammonia that dissociate from the amino acids are toxic. For where? For our brain. The brain have high rate of metabolism, and the production of ammonia in brain is high. So, if the ammonia released to blood circulation freely, it is dangerous. What is happening? The ammonia enter the glutamate. Do you know glutamate, an amino acid?
Yes.
And convert it to another amino acid under the name glutamine. This ammonia entered to a structure of amino acid. Now it's not dangerous anymore because it is in amino acid structure, especially in brain or skeletal muscle or skin or liver or central nervous system, everywhere. Now this ammonia is in a non-toxic way and go to blood circulation and enter where? Liver. For what?
Because the pancreas will release the enzymes to break down the proteins or something.
No, this is amino acid, not protein.
The same mechanism of lipid. When they enter the liver, there is an enzyme that convert to provate, and provate.
In some situation, the amino acid that are glucogenic exactly convert to pyruvate. But in liver we have urea cycle. You know, the amino acid can remove the ammonia from glutamine and convert it to glutamate, and this ammonia enter the urea cycle. The liver is the place that urea cycle occurs. You should memorize this point. I will ask you that which organ is the place of urea cycle? Kidney, liver, brain, muscle.
No.
Aziza, liver. Liver, guys, be careful about these points that I am mentioning for your final exam, okay? The liver is the place that urea is produced and excreted from our body. As our friend mentioned, this glutamate and maybe this amino acid convert to pyruvate and glucose. I will explain a little bit about them. It is the way of excretion of amino acids. Guys, this is urea cycle. I don't want to memorize none of them. Just I want to know you that what is happening here. You can see here that ammonia comes from liver. Can you see? The ammonia, this is NH4. Okay? And on the other hand, another ammonia come here and produce urea. The urea excretes two nitrogen from our body. Okay? Just I want to know you that urea cycle occur in the liver for what? For production of urea. For what? To excrete the ammonia from urine. Guys, all of you understand the points or not?
I don't understand the urea cycle very much.
You didn't understand?
Only the urea cycle.
The urea cycle, do you want to explain all of these or not? Sure not. Okay, the urea cycle, I told you. We have, wow.
Zoom it if you want.
We have ammonia here, am I right?
Yes.
This ammonia enter the urea cycle. This ammonia enter the urea cycle.
Okay.
What is urea cycle? Urea cycle is a cycle that occurs in which organ? Liver. In which organelle? Mitochondria and cytosol. In these two organelles, cytosol and mitochondria. These ammonia enter the reaction and produce some substances. After that, another ammonia comes from another amino acid under the name aspartate. With consuming lots of energy, the ammonia is produced. The urea is produced. To what? To carry nitrogen out of our body through urine. Did you get the point or not?
Yes.
What about other friends? Do you want to your friend explain you in Arabic? Yes or no?
We got the point.
Got the point? If you want, we can ask your friend to do that. Hi. About amino acids, if you remember, I told you some of amino acids are glucogenic, can produce glucose, and some of them are ketogenic. They can produce acetyl-CoA. They can produce ketone bodies. Ketone body is produced from acetyl-CoA and during the fasting can produce energy for us. Guys, have you ever seen that during fasting in Ramadan, Shahr Ramadan,
Yes.
in Shahr Ramadan, our mouths have a bad smell like acetone. This is related to the situation that our body starts to breaking down the lipids, and in those situations, the ketone bodies, acetoacetate, beta-hydroxybutyrate, and acetone is produced. Acetone exhales from our lungs, and that's why our mouth and our breath have a bad smell. It is related to the metabolism of fat in our body, lipids in our body. Okay, some amino acids guys, this is the situation of fasting, not in normal situation. Amino acids convert to glucose precursor or ketone body precursor. That it's not important to memorize this slide, but I want to tell you which part you should memorize, guys. One of the best thing that I can ask you is about the ketogenic amino acid. Please, instead of question bank, write it down in one place. I will ask you which of them is an ketogenic amino acid. You should know lysine and leucine. It doesn't need to memorize all of them. Just now, leucine and lysine are ketogenic. Okay? Do you want to tell them to Arabic? This one? It is okay? Okay, memorize that leucine and lysine is ketogenic. Other them can produce glucose, and some of them can produce both glucose and ketone bodies with each other. Guys, memorize this and have a rest and come back to the next